Okay, I think we'll go ahead and get started with our next workshop on breaking investigative barriers with DNP. I'll turn it over to Krishnamurthy to get us started.
Thank you, Eric. Yeah, I'm essentially stepping in for Dave Rice today. Good morning, everyone, or good evening, everyone. Today, we are joined by Ruth Stark and her team. Professor Stark is from City University of New York. I have known her for many, many years by now, and she is trying to. We are one of the items that we were talking earlier today was trying to break the barrier between different areas of NMR, solids and liquids and imaging. And the idea is we learn from each other technology. And I really look forward to today's workshop. And I will let Ruth introduce her panel and take it away. Take it away, Ruth.
Okay, thank you, Krish. And good morning or afternoon or evening as the case may be. I think the theme today is a theme that is familiar to all NMR spectroscopists. That is the battle to improve sensitivity. And we are constrained, but not completely constrained by the physics of our NMR spectroscopy. And the idea here is to consider some of the most challenging, namely the solid state problems, and to talk about a technique that's been around actually in the original form since the 1950s, but in the modern form maybe since the early 1990s, and that's dynamic nuclear polarization. And what I've done here is to recruit three experts in the field who study different kinds of materials, either biological, natural, or engineered, and let them tell you their take, each of them, on this situation. Now, I have to say, when we thought about this, we always have to think about our audience. And our audience is interested scientists who may or may not have a lot of experience in DNP. And I am one of those. I think my group has published a few DNP spectra, and we have worked on the technique, but we are far from being expert. And in my case, I am about 150 yards from the New York Structural Biology Center, where one of maybe the first in the US was installed. And one of the people who used to run the advisory committee used to run into me on the subway, and he would always turn to me and say, "This DNP stuff, is it good for anything?" And we had 7 minutes on the subway for me to make the elevator pitch, except it was in a subway, to speak to that issue. And we are still speaking to that issue, I think. So I invite you to listen and to give us feedback. The chat is open and we will be monitoring there, but I think by and large, we will take questions at the end unless there is something that we feel needs to be addressed before the next panelist takes his or her turn. And I hope you will enjoy it. So the first of our panelists is Aaron Rossini. He is a professor of chemistry at Iowa State University, and he's going to tell us about his favorite materials. So Aaron, the floor is yours.
Okay, so I'm going to try to tell you a little bit about how we can actually do magic angle spinning dynamic nuclear polarization solid state NMR experiments to look at the surface structure of inorganic materials. I tried to make this more educational rather than really covering all the kind of examples out there. I wanted to give people an idea of how we actually do these experiments. Before I get into the actual data, I just want to thank the group members. So I think most of the work I'm going to show you today was done by two former students in my group, Dr. Yunhua Chen, she really led the project on the nanocrystals that I'll tell you about, and Dr. Rick Dorn, he did the work on the boron nitride that I'll tell you about at as the first topic. And all of this work was funded by the U.S. Department of Energy, Basic Energy Sciences, as part of our research program at Ames National Laboratory. So my group is really interested in materials to address energy needs. So different types of systems that we're interested in studying are semiconductor nanoparticles, semiconductor nanocrystals, 2D materials, and heterogeneous catalysts. And if we think about the kind of rational development or design cycle for these materials, usually chemists or material scientists are going to be synthesizing materials, measuring properties. Nowadays, there's going to be some sort of computational modeling or prediction involved. And then we ultimately need to perform molecular level characterization to actually understand what was made and how we can tie this back into the properties. But characterization is often a real challenge in this class of materials. So many of these materials are disordered, they're not crystalline, or it could be that their activity is really being driven by dilute sites or defects. And then the properties are often controlled by the surfaces or interfaces. And so in general, the characterization of surface species is a real challenge in science. And it's an unresolved challenge. We don't really have general methods for this. So one of the things my group is interested in doing is using multinuclear NMR experiments to characterize materials. And so solid state NMR is really an ideal characterization technique for materials. We can really get a lot of structural information. So we can look at the local structure and symmetry when we measure chemical shifts or quadrupolar coupling constants when we deal with quadrupolar nuclei. One advantage of doing solid state NMR is, in addition to measuring the isotropic chemical shift, we can also measure the anisotropy of the chemical shift. So I'm just showing this example of this platinum 195 solid state NMR spectrum of cisplatin that was obtained in my lab. And by looking at the CSA, we can tell this is a 4-coordinate platinum site. And the isotropic chemical shift is telling us something about the identity of the ligands that are in the first coordination sphere here. When we measure quadrupolar coupling constants, those tell us a lot about the symmetry at the nuclear site. So I'm just showing this example of boric acid versus a tetrahedral boron site. When we have a trigonal planar 3-coordinate boron, we get a sizable quadrupolar coupling constant. We have a tetrahedral boron, there's negligible quadrupolar broadening. And then really, of course, the power of NMR is the ability to perform 2D NMR experiments that can elucidate bonding and connectivity. So this is a DNP-enhanced cadmium, selenium, JHMQC spectrum, and I'll tell you about this one a little more later on in the talk. And then really another powerful aspect of solid-state NMR is the ability to perform interatomic distance measurements with precision that rivals that of X-ray diffraction. So this is just an example from my lab where we were able to measure the distance between this alkene proton on this cyclooctadiene ligand and a platinum center by performing this proton platinum Redor experiment. So in general, when we deal with inorganic materials, we're not going to just deal with carbon, nitrogen, hydrogen, or phosphorus, the kind of common nuclei that we encounter in organic systems or biomolecules. We're going to really be dealing with whatever elements or nuclei are present in the materials that we're given to study. So one of the challenges of this is that a lot of the elements have properties like large quadrupole moments, low frequency, low abundance that make it challenging to study them. And so this is where we need to come up with sensitivity enhancement techniques. So in my group, we like to use things like ultra high magnetic fields, DNP, and indirect detection schemes to enable the studies of these unreceptive and exotic nuclei. Okay, so let's try to get back to DNP. And first, I'll give you a little bit of motivation. So this is just one case study I wanted to point out here. Hexagonal boron nitride is a material that we got interested in studying because we were working with a collaborator who was using it as a catalyst for the dehydrogenation of propane to make propylene. And so when we started looking into the literature about this material, HBN, if you're not familiar with it, it has a very analogous structure to graphite. You have this honeycomb lattice, it makes these atomically flat two-dimensional sheets where you have alternating boron and nitrogen atoms. But for catalysis and for a lot of material science applications, it would be important to understand what is the surface structure, what is the structure of these atoms that sit on the edge of this material. And so when we looked in the literature, there's some nice TEM studies where you can maybe start to image the edges or surfaces of these materials. But then there's lots of computational studies that suggest various oxidized edge terminations for this material. So this is one of the questions we wanted to answer. So the way that we made the boron nitride materials for study in this, or that we studied by solid state NMR, is we use the simple solution exfoliation procedure. So what you can do here is you can take bulk boron nitride powder. This is an SEM image of the stuff you would get out of the bottle from a commercial vendor. You can just sonicate it in water, centrifuge it down, and then there's going to be some fraction of the boron nitride that's colloidally stable and remains suspended in the solution. We can then collect this solution, evaporate the water, and see what's precipitated out. And when we look at now a TEM image after this exfoliation procedure, we've got much smaller boron nitride flakes. Again, these materials have this 2D or flake-like structure because inherently they're a two-dimensional material that's made up of these atomically thin sheets. And so on average, after we go through this exfoliation, these materials are reduced to about 20 to 50 layers in thickness, okay? So these are now small enough kind of nanocrystals or nanoparticles that we can do NMR experiments to look at the edge structure. Okay, so what's the challenge though? Even if I have a 100 nanometer by 100 nanometer boron nitride flake, and I want to look at the structure of the atoms that sit on the edge of this sheet, okay, we can run some simple statistics or count the atoms. There's about 3,500 edge atoms out of 390,000 total atoms. So we have this problem that only 1% of the atoms actually reside on the edge, and that's the atoms that we want to understand the structure of. So right away, there's going to be a 110-fold reduction in sensitivity if we could do a hypothetical edge selective NMR experiment. And this is really problematic because, as you're probably all aware, the experiment time goes with the square of the change in the sensitivity, so 12,000 times longer experiments. We have another problem. We need to come up with an NMR experiment that's selective enough to see the atoms on the edges, okay? So we have this dual problem of sensitivity and selectivity. And so the way that we can address the sensitivity problem is by using DNP. So really the problem with NMR is that the energy differences for the spin states in the magnetic field are small as compared to the thermal energy, so as compared to KBT. And so consequently, we have very small polarization or negligible polarization of our nuclear spin states. The upper and lower energy levels are almost equally populated. And this is why we need to have these big magnetic fields, work on large sample volumes, and do a lot of signal averaging when we do NMR. But if we look at a different type of spin, unpaired electron spins, those are much more polarized in a magnetic field. So for the 263 gigahertz, 9.4 Tesla DNP spectrometer that we use, the electron spin polarization is about 6%. So this diagram is exaggerated, of course, but it's just to illustrate that the electrons are much more spin polarized. So the idea of a DNP experiment is we're going to dope or introduce our some stable free radicals that have electron spins into our sample. And then we're going to do a double resonance EPR and NMR experiment. So we can irradiate the electron spins at their correct frequency, which for 9.4 Tesla field is 263 gigahertz. Then we can actually transfer spin polarization or population difference to our nuclear spin states. And we can get a massive enhancement in the sensitivity of our NMR experiment. So in my group and lots of the other groups that are working on magic angle spinning DNP, we're using this commercial Brooker DNP spectrometer. At the Ames lab, we have a 400 megahertz, 263 gigahertz spectrometer. This is a picture of the actual setup. And then here's a more schematic drawing that you can see here. So this system was really developed by Dr. Melanie Rosé, who's unfortunately no longer with us, but she made really great contributions to the field of DNP by making this commercial instrumentation available to everyone. And it's really, this instrument is really based upon the design of Griffin's group at MIT. So what you have in these magic angle spinning DNP spectrometers are three key components. We have our 9.4 Tesla NMR magnet. I think Brooker's selling now up to 21 Tesla, however. But you have your NMR magnet. That's like a conventional wide bore NMR magnet like you'd use for most solid state NMR experiments. And then we have an MAS cooling system, which generates nitrogen gas that's cooled to about 110 Kelvin that we're going to use for the magic angle spinning experiment. And then we have the gyrotron over here. The gyrotron is one of the few devices that can produce high power at this frequency of 263 gigahertz that corresponds to the Larmor frequency of the electron spins in this 9.4 Tesla magnetic field. So the gyrotron puts out about 10 to 20 watts of power at 263 gigahertz. Okay, so how do we do a DNP experiment? We take the material of interest, we dope it with the free radical. So in this case, I'm just showing this is microcrystalline histidine hydrochloride. We ground the histidine into a fine powder. and then we impregnate it with a solution of this biradical TechPol. The solvent that we used to disperse the radical was tetrachloroethane, and the radical concentration in this case is about 15 millimolar, so it's pretty dilute. So typically you'll take something like 30 or 40 milligrams of your ground material, add anywhere from 5 to 20 microliters of the radical solution. You'll get this wet powder, you'll pack it in the rotor, and then you'll put that rotor into your DNP system, which is all cooled down to about 100 Kelvin. So once it's inserted into the DNP spectrometer, everything's frozen. Okay, so on the right here, I'm just showing some proton-carbon cross-polarization magic angle spinning spectra that we obtained without DNP, so the gyrotron is turned off. Or we can go turn the gyrotron on, continuously irradiate the sample with 263 gigahertz microwaves. What that does is it drives a transfer of spin polarization from these unpaired electron spins to the proton spins that are residing in the histidine crystals. And then we can transfer that proton spin polarization to the carbon spins via cross polarization. And so when we do this, we can increase the intensity of the histidine NMR signals by a factor of 150. We call that the DNP enhancement. And now we go from a signal to noise ratio of 15 after 8 scans to a signal to noise ratio of 2,100 in 8 scans. And this is natural abundance carbon-13. So really, this is the motivating factor to be doing these DNP experiments. We can really get these sensitivity gains that really change what you can do with NMR. Okay, so coming back to boron nitride, this 2D material, we wanted to understand the structure of the atoms on the edges of this material. How do we do a DNP experiment here? Well, we take our boron nitride particles, which I've kind of represented as the stacked sheets. We impregnate it with a solution of our polarizing agent. In this case, we're using TechPol, which is a biradical polarizing agent that's optimized to work well at 9.4 Tesla. So now we're going to have some polarizing agents that sit near the atoms on the edges of these 2D sheets. And remember, this is all frozen at 100 Kelvin. Okay. We turn on our gyrotron. We send some microwaves in that will irradiate all of the electron spins throughout the sample. This will initiate a polarization transfer from the electron spins of the radical to nearby proton spins. There would be solvent here as well, but I'm just showing the hydrogen atoms that sit on the edge of these boron nitride sheets. And there's a lot of proton-proton spin diffusion that happens in the solid state, which can kind of uniformly mix this DNP-enhanced proton polarization throughout the sample over nanometer length scales. And then at this point, we're now ready to do a surface selective NMR experiment, which we usually achieve just by doing cross polarization. So if we do cross polarization to the boron or nitrogen spins that reside on the edge of the sheet, it's really the cross polarization that's providing us with the surface selectivity, because CP is really going to only be effective over length scales of a few angstroms. So this is what's going to allow us to observe the NMR signals from the atoms that reside on the edges of these materials or on the surface of the material. And I should just point out, this is really the DNP Sense dynamic nuclear polarization surface enhanced NMR spectroscopy approach that was developed by Anne Lesage and Lyndon Emsley back in 2010. And initially they demonstrated on silica. I'm just showing it for this 2D material for illustrative purposes. Okay, so with DNP now, we can do a natural abundance nitrogen-15 NMR experiment overnight on these boron nitride sheets. The experiment time here was 17.5 hours. On this particular sample, we got a DNP enhancement of about 200, which roughly corresponds to over a 50-fold improvement in sensitivity. And so without DNP, this type of experiment would have taken more than 4 years of signal averaging, which obviously is just not feasible. Or, of course, we could isotopically enrich our materials if that's a possibility. But really, the ability to work with natural abundance materials is a key benefit of DNP. Okay, so when we look at this nitrogen-15 NMR spectrum now, we have 4 signals that are present. This signal at about -280, that's what you would see if you did a direct nitrogen-15 NMR experiment on boron nitride. Those would be the nitrogen atoms that reside in the center of those 2D sheets. But we have these additional signals. How do we explain those or interpret this spectrum? What we do is some plane wave DFT modeling. We make some simple slab models where we can terminate the edges of the HBN sheets in different ways. So these we call the zigzag edges of hexagonal boron nitride. You can see why. So 1/2 of these sheets are nitrogen terminated, and each nitrogen atom is terminated with a hydrogen atom. The other half of this sheet is boron terminated. We have a hydroxyl group that caps each of the boron atoms. We can also have an armchair termination. You can see why this is called an armchair, perhaps, where we have alternating boron and nitrogen atoms along this edge. again, we have an H atom termination of our nitrogens, and then our borons are hydroxyl capped. And what's interesting here is if we look at the DFT calculated nitrogen chemical shifts, we can see excellent agreement with the experimental NMR spectrum. So by doing these types of surface selective NMR experiments, combining it with DFT modeling, we can really come up with these atomistic models of what the surfaces or edges of materials look like. We also did a boron NMR experiment with DNP. So the way that we did this experiment is we do DNP enhanced proton to boron cross polarization step, and then we added a spin diffusion filter here. So whatever magnetization initially gets generated off the cross polarization step, we store that along the Z direction. We let the boron nuclei spin diffuse. Boron's about 80% abundant, so spin diffusion is relatively fast. And then we can read the magnetization after the spin diffusion period. So if I do a short spin diffusion period, I get this broad boron signal. We don't have the best resolution because we're only at 9.4 Tesla, but believe me, this is corresponding to boron that's bonded to oxygens, okay? And then as we increase the boron spin diffusion time, we can see that the signal shifts more to the left. Boron nitride has a shift at about 30 ppm. So what we think is happening is we're starting on these oxidized boron edge sites, and we're going into the boron that makes up the core of these sheets, okay? So this shows us these are not just impurities, these are actually part of the material that we're studying. Okay, last application, semiconductor nanocrystals. You're probably familiar with this because this was awarded the Nobel Prize in Chemistry in 2023. So in a nanocrystal, we're gonna have an inorganic material, which is usually a semiconductor, and it's gonna often be capped or terminated with organic ligands. The organic ligands prevent the aggregation of the particles and prevent them from forming larger crystals. And by controlling the synthesis, you can control the size of these crystals. And what's important is when you change the size of these semiconductors, you can change their band gap, and that allows you to tune the color. So these materials have lots of applications in displays, photovoltaics, and biological imaging because you can really tune the luminescent colors of these nanocrystals. But what becomes really important when you deal with nanocrystals is because they have a very small particle radius is often on the order of 1 to 10 nanometers, you end up with a very high surface area to volume ratio. So in other words, a significant fraction of the atoms that make up these nanocrystals end up being surface atoms. And the surface atoms have the structure of the surface atoms really affects the bulk photophysical properties of these materials. So this is a nice example from John Owen's group at Columbia, where they're showing what happens when you strip off the carboxylate ligands that cap this nanocrystal at high carboxylate ligand densities where the surface is well passivated. You get highly luminescent materials, but as you strip off these ligands, you lose the luminescence of your nanocrystals. So this is just underscoring the impact that surface structure has on the bulk properties of these materials. Okay, so that's the motivation to do DNP. We can again look at the surface structure of these materials. One of the things my group worked on was better ways to formulate nanocrystal samples for DNP experiments. So back in 2015, I was fortunate enough to collaborate with Maxim Kovalenko's group when I was a postdoc with Lyndon Emsley. And we came up with this method for doing DNP NMR experiments on nanocrystals. So the idea is we would take our nanocrystals, which are usually colloidally stable in solution. We could take that nanocrystal solution, impregnate it onto a mesoporous silica, and then we would add our radical polarizing agent solution at the same time. And so what this would give us is silica pores that are filled with both nanocrystals and our polarizing agent. The reason we had to do this is if you just try to add the polarizing agent to a nanocrystal solution and freeze the nanocrystals, they just precipitate out to the bottom, and then you get phase separation of your nanocrystals and your polarizing agent. So by trapping them in the pores of the silica, when you freeze the sample, you prevent them from separating, and then you get decent DNP enhancement. So this experiment works pretty well. We can get a DNP enhancement of about 40 for the ligands on the surface of the nanocrystals, and then we can get a cadmium spectrum of these CAD sulfide nanocrystals in only a few minutes. One of the things that we found worked better though, and one of the problems with this method is our nanocrystals are very dilute. So now oftentimes what we do if we're going to do DNP experiments on nanocrystals, we just collect precipitated nanocrystals. They're kind of just like a bulk powder. We mix that powder up with a HBN, hexagonal boron nitride. The reason we use hexagonal boron nitride is it helps to focus the microwaves into the sample better. And so this results in a higher DNP enhancement. And because the nanocrystals are more concentrated now, they're making up roughly 50% of the sample mass, we overall get much better sensitivity enhancement. So if you compare the cadmium spectrum that we got by this sample preparation method to this one, you can see the dramatic improvement in the cadmium sensitivity. It's about a factor 9 better. So just trying to underscore here, whenever you do a DNP experiment on a material, you really have to think carefully about how you formulate it. And the sample formulation is often a key component in getting DNP to work. Okay, so what can we do with DNP? This will be just 1 or 2 slides here, Ruth, then I'm done. So here I'm just showing the cadmium solid state NMR spectra on the left, selenium solid state NMR spectra on the right. And these are for cad selenide nanocrystals. Cad selenide is one of the, cadmium selenide is one of the kind of canonical nanocrystals that is out there in the literature. And so we've made these nanocrystals with 2 different shapes using some different synthetic recipes. So we had plates, okay, those spectra are shown in the pink here. And then we had spheroids, those are the more kind of purple or cyan color that you're seeing there. What's interesting is when we look at the cadmium and selenium spectra for the plates and the spheres, we see that those spectra are very similar in appearance. These are all spectra that we obtain with DNP, and then we do cross-polarization from the protons that are on the surface ligands of these crystals to the cadmium or selenium atoms that sit on the surface. So predominantly we see surface signals, but we can also see NMR signals from atoms that sit just below the surface that are in the more crystalline region of these particles. So we got DNP enhancements here of about 3785. And so what this means is now we can get these 1D cadmium or selenium NMR spectra in just 10 to 30 minutes. That's not such a big deal because you could probably get these 1D NMR spectra using conventional NMR in an overnight experiment. But by getting the experiment time down to 10 minutes, then you could really do experiments that would have been impossible previously. So what we were able to do, because we had DNP working well on these cadmium selenide materials, we were then able to actually do a selenium cadmium JHMQC experiment. So it turns out cadmium selenium 1-bond J couplings are about 100 hertz. So we were able to record this 2D JHMQC. And so what we can see are the selenium atoms that sit on the surface of this crystal. They connect to the surface cadmium atoms as we would expect. And then we can see that those surface seleniums connect to core or bulk-like cadmium atoms. And then we can see that there's connections between core cadmium and core selenium atoms. We're missing the core surface connection because that's more than 1 bond away. So really what we're kind of doing here is a sequential walk to build up the structure of these nanocrystals. We can also do some more advanced experiments. This is a J-resolved experiment where we look at the extent of dephasing while we observe the selenium signal, while we apply a pi pulse on the cadmium channel. And so by fitting this curve, this confirms that our surface cadmium atoms are bonded to our surface selenium atoms are bonded to 4 other cadmium atoms. That's the main piece of information we get out here. And then we can reverse this experiment. We can look at the surface cadmium NMR signal while we pulse on selenium. And by fitting the height of this dephasing curve, we can confirm that each of the surface cadmium atoms is only bonded to 2 selenium atoms. So this kind of validates these structural models that we're able to put forward. All right, well, I think I've taken enough time. I just quickly threw this slide together too today, just to give some things maybe for the discussion period, but DNP is really a new technique. Commercial instruments have really only been available since 2010. There's lots of groups that are working on developing different aspects of DNP, and these are really going to enable further advances in the science that we're able to do if we want to look at things like materials, catalysts, and pharmaceuticals. So, high field DNP, helium, MAS DNP, improve microwave devices that can do pulse DNP, improve pulse sequences so that we can study nuclei. So we need pulse sequences that let us transfer magnetization from protons to whatever nuclei we want to study. And they need to be applicable to quadrupolar nuclei and nuclei with large CSA that we encounter in materials. Improved radical polarizing agents. Another really interesting idea, maybe we can get rid of the gyrotron in the future and just use light to photoexcite polarized electron spins that will spontaneously do DNP on their own. There's been some really exciting work along this direction by a number of groups. DNP with endogenous radicals, so radicals that are just intrinsic to materials, and then combining DNP and fast magic angle spinning is an ongoing goal as well. One last thing, I'll just say, I didn't talk about pharmaceuticals. I've also done a lot of work on getting DNP to work for pharmaceuticals. If you're interested in pharmaceuticals and DNP, you can go check out this 2018 magnetic resonance and chemistry review that I wrote. And with that, I'll I'll say again, thank you to the students who did the work and turn it back over to Ruth.
Thank you very much, Aaron. What a tour. What a tour.
As always, give a word for our sponsors. First, MR Resources, they've been around for 39 years at this point, servicing the international NMR community and doing pretty much anything and everything that needs to be done with your NMR system. Magnet quench recovery, magnet and system moving and relocation, service contracts, reconditioned NMR systems, reconditioned magnets. And it's always very important for me to point out that they have absolutely a full supply of spare parts for everything that they sell. And they have engineering expertise that I dare say is second to none. Give some thought to doing business with MR Resources.
What I'm going to do at this point is to turn things over to our second panelist for something entirely different, I think. That's Tuo Wang from Michigan State University, where he's associate professor of chemistry. And I think he's going to tell us about plants and or fungal cells, but it's up to him. Go ahead, Tuo.
Yeah, thank you, Ruth. Yeah, so I really enjoyed the lecture by Aaron, and he also covered a lot of aspects on the DMP methodology part. So for me, I'm not a DMP expert. Yeah, so I'm basically using this technique for different applications. And here I will show you 3 ways I use the DMP to address 3 different types of questions. So I will keep this part short, but from the view of application scientists, for me to understand DMP, when we have the radicals doped into the materials, we have microwave irradiation, which will polarize the electrons and then transfer the electron polarization, for example, through cross effect for the biradical to the protons in the solvent and then to the protons in the bound molecules, and then finally transfer to the carbon-13, nitrogen-15, or other nuclei in the bound molecules. And the favorite radical we are currently using is this AC ProPork, which is a very good sensitivity enhancement and also give very fast relaxation and also fast DMP build-up time, which means your scans can be collected in a faster way, and then your experimental time will be shortened also because of that. And because DMP requires special instrumentation, and here is the illustration made by my previous student, Alexander, and it's basically showing the setup of the DMP system at my lab. I don't have DMP in-house, so I always travel to my lab to use their instrument. And we need this cooling cabinet to cool down the temperature to around 100 Kelvin for the sample, which also brings some issues, which I will show later. And then for the DMP setup at my lab, they have this kind of waveguide, which can split the microwave from the gyrotron into 2 beams. One sent to the MS DMP, which is a solid, and the other one sent to the dissolution DMP or some other DMP. I forget the exact type. But through my collaborations with Fred Montink-Wigner in the past years, he always educated me to mention MS DMP specifically because there are so many different types of DMP, and this is the one that we're using here. So I will start with some questions that I typically receive. Yeah, basically for every presentation I make, I will receive those questions. The first thing is, how could you make sure that you are giving homogeneous DMP, homogeneous polarization for all molecules in your sample? Well, if we think about sample preparation, we dope the radical into the material, and for the materials handled in my lab, they are porous. Yeah, so with the pores typically on the range of tens of nanometers. So they are large enough because the radical itself only have like a dimension of 1 to 2 nanometer across typically. And then the radicals can penetrate into the porous materials, and then they can rely on proton-proton spin diffusion also, so for transferring the polarization further and further, so after that. And the easiest way to check the polarization is just to overlap your microwave on and microwave off spectrum, basically with and without DMP, to see whether spectral envelope is kept the same. If they show similar patterns, then it means, okay, molecules in your spectrum, they're polarized in the same way, homogeneously. But we do met many systems where the molecules are polarized differently. For example, typically for aromatic polymers, they receive lower enhancement compared to carbohydrates. Yeah, that's very common because whenever you have molecules that cluster together to form nanodomains that are beyond the tens of nanometer range, they will not receive perfect DNP enhancement. Yeah. And another question is, okay, whether you're, because we are doing experiments at a low temperature, then typically the lines are much broader than at a high temperature. For example, you can see that comparison of the DNP spectrum with the room temperature spectrum, all the sharp peaks are gone, while the broad peaks from cellulose are retained. Then the question is whether this is caused by the low temperature effect or is caused by the paramagnetic relaxation enhancement, the PRE effect, due to the doping of the DNP radical, the biradicals. So that can be easily checked by just overlapping samples prepared with and without the radical. You can see that the long widths are roughly the same, so between those samples, or sometimes they may be different. Yeah, if they're different, then it means that you have too much radicals too close to your molecules. Then basically, reducing the radical concentration a little bit will help. And another thing is that if you have mobile and rigid molecules present simultaneously in your sample, typically the more ordered molecules will keep their long width at a low temperature, and the more mobile molecules will be broadened out because they have very broad distribution conformations. And then when all in sudden your sample is frozen and bring to 100 Kelvin, then their broad distribution conformations will be kept. Then they will have very broad long width at the DNP condition. So that's some downside of the DNP. So now I will talk about the three applications. The first thing is to visualize the polymer packing interface using label samples. The second one is to resolve unique structures of a molecule from the bulk equilibrium, although they have the same chemical structure. And the third one is basically natural abundance DNP explored by the POPs group, and we're following their efforts. So here I will show you some cartoon illustration of the plant samples being studied in my group. And here they're basically very heterogeneous and complex materials formed by lignin, which is aromatic polymer, and many different types of carbohydrates. For example, they have the fibrils here, white fibrils, which are cellulose. They have the red band, which is the xylene in flat ribbon conformation, and then they also have twisted conformation, the blue bands. Then the question we want to address using DNP is, okay, how those different polymers are packed together, especially how lignin pack with carbohydrates. Yeah, which carbohydrates it will bind, and which conformation it will bind. So this will require the DNP to help us, because if you think about the equilibrium state, the carbohydrates are much higher than aromatic polymers. Yeah, so it's more than 100-fold stronger in signal. If you measure two-dimensional carbon-carbon correlation experiments just using regular way, it will be a mess. Yeah, the carbohydrate will dominate and its side bands will dominate. So with the DNP, we can get like a 23-fold sensitivity enhancement on the plant samples. It's not perfect, but it's sufficient. And then we can apply different kinds of aromatic selection to select the lignin signal against the carbohydrate signal. And then we transfer the lignin polarization to carbohydrates to measure two-dimensional correlation experiments and compare the lignin bound portion of carbohydrates with the bulk equilibrium. And you can see that the cellulose signals get either disappear or get impaired, while xylene signals, they are retained in the lignin bound portion of the carbohydrates. And specifically, we can even distinguish different conformations of xylene, and we can show that the flat ribbon structure is binding surface of cellulose microfibril, while the non-flat structure is binding lignin specially. So here's the illustration of the cell wall structure of the fungal cell wall, so which is when I started those studies, my research is largely inspired by Dr. Ruth Stark's research. So I read her papers and decided, we want to apply DNP on this. So for these systems, they have proteins, they have mannan, they have galactan, they have alpha and beta glucan. And for the beta glucan, they have 1,3,1,4 linkage, 1,3,1,6 linkage, and 1,3 linkage, all different kinds of covalent linkage. And they have chitin. Each single type of polymer here will have special NMR signal, and we want to correlate them. It's many cross peaks. Yeah. So if you think about those studies, typically we combine solid-state NMR high-resolution data, mobility, hydration, and conformers with the DNP data of the intermolecular packing, long-range correlation cross peaks, and with the chemical assay and the imaging data. Combining the information will give us some vision of those complex materials. For example, we got 17-fold sensitivity enhancement on the mycelia or aspergillus fungus, which is not perfect, but once again is sufficient, and will allow us to measure those noiseless spectrum with short range and long range correlation. Each new signal showing up in the gray spectrum, they represent some nanometer carbon-carbon correlation between two molecules. We can assign them, which will be time consuming, and then we can restrain the polymer packing interface and further feed those information to MD modeling. So for the next step, which we have done recently in this study. The power of DMP here is, so you have so many different molecules. Each of them only account for 5 to 10% of the sample of everything you can pack into the rotor. And then you have like 7 to 8 different types of molecules, and you can observe all the interactions between those molecules. And that's basically based on the sensitivity enhancement of DMP. So now we will talk about second application to resolve molecule from the bulk equilibrium with the same chemical structure. This story actually started from the proton detection, where we observe 3 different lines of beta-1,3 glucan signals, so with a different chemical shift. So the chemical shift difference is very small, it's like 1 ppm. If you look at the carbon-3 site, and then we want to correlate the MR signal multiplicity with their structural difference and also function difference. Yeah. So then we dope acid-pork into the material, and for those yeast, they are unicellular. Each cell is by itself. So we get much higher sensitivity enhancement. We get 57-fold sensitivity enhancement because of better mixing with the radical. And then when we look at the DMP spectrum, here we show the 5 millisecond par proton acetyl recoupling spectrum. You can see the dominant signal here, which correlate with the proton detection signal here. And then you can see a minor form, so 1 ppm apart for the carbon-3 site, which is correlated with the type B signal here. Yeah, and then when we correlate with the chitin signal, then you find only this minor form is correlating with chitin. Yeah, so it basically means that the bulk equilibrium of the beta-1,3 glucan in the model triple helix structure is not interacting with chitin, and only this minor form, so here and here, they are packed with chitin microfibril, and because of this kind of packing interaction, their structure has been changed. They no longer can adopt this kind of triple helix structure, but they have to be flattened in some way to allow this kind of interaction to happen. And at the same time, we know that chitin and microfibrils, they have very flat surface, and then this will allow the molecule to be docked on the surface of the flat region. So the last thing and the most exciting thing is the natural abundance DMP. This part is not by me, it's basically by Dr. Gail the Pope and many other researchers, and we're just following their track and to do some applications on our sample. And why we want to do natural abundance DMP is because many different samples, they cannot be isotope carbon-13 labeled. For example, we have some fungi that can only be co-cultured with the cells. And then for some other fungal species, we need to select them from like for certain morphotype. For example, we want to have clean conidia rather than mycelia, and rather than mixture of different cell types, then we can only handle those unlabeled materials. And here, our collaborator, Song-Soo Wee, has demonstrated that the double quantum filtered DAR experiment is very powerful for the natural abundance DMP or unlabeled materials. And you can see the noiseless spectrum collected on 30 milligram of unlabeled conidia within 30 hours. And you can see the beautiful protein signals, you can see the carbohydrate signals, and the closer look of carbohydrate region will allow us to resolve different carbohydrate components, including chitin and beta and alpha linked glucans. Although the resolution is no longer that sharp as in room temperature condition, at least we can resolve some molecules and get some structural information out there. And another application is to the plant side. So during a trip I had back in 2020, I believe, so I met with agricultural folk. He bring me to the field. Yeah, so they show me some like a red stem with like a normal phenotype and some other red stem with a very brittle phenotype. We cannot label them. They have been grown for like 5 months or 4 months. And then we just bring those stems back to the lab, and then we measure the DMP spectrum, and with 35 hours, and then we can measure those very high resolution, beautiful DMP enhanced spectrum, carbon-carbon correlation, and compare them with the label stem, and we can further compare the wild type with this kind of brittle mutant. And you can see the beautiful signals of all glucose environments in cellulose. and also see the twofold xylan and threefold xylan, although their signals are slightly weaker than room temperature. However, you should realize that some signals disappear, especially for the pectin signals here, because they are so mobile. They're not structurally ordered. They don't have any structural order. They're just like in solution. So their signals are broadened out completely. And recently we combined DMP with Core-EM. So Core-EM gave us the information on the nanoscale for the carbohydrate components. They are not as beautiful as the proteins. Yeah. So for here you can see two bundles are wrapped together and they are twisting. You can see different cross sections, and you can see the density map of the electron. And the DMP allows us to resolve the atomic level resolution and restrain the structure into an 18-chain model, which can fit into the nanoscale structure provided by the Core-EM. And here's a strategy how we combine DMP with other biophysical techniques, so to bridge different length scales. And I believe it can be applied to other biomaterials as well. And we are going more and more wild. Recently, we start to work on the soil. And we look at some wetland soil collected just 1 hour away from New Orleans. And our collaborators, they just land a boat on an island and collected the soil material directly from using a pole from 2-meter depth underground. And by carbon-14 dating, so the soil sample, their age is from nowadays all the way to more than 11 centuries, so backward. And then we measure this 1 hour, so proton-carbon head core experiment. If you look at the sensitivity and resolution, it's just perfect. And if you want to sacrifice some sensitivity, we can even further cut it to 20 minutes per sample if you want. Yeah, you can see the beautiful signals, so cellulose and the xylan, threefold and twofold xylan. You can see the methyl esters, you can see the lipid chains, and you can see aromatic polymers, so basically lignin, all of them. We can do assignment, and we also measure the carbon-carbon correlation experiments on those, which we didn't show here. And furthermore, we can correlate them with each other to detect their long-range correlation, how they are packed in space. And this can also be finished within a single hour. And here you can see that the carbohydrate protons, they start to show cross peaks with aromatic carbons. Yeah, so for here we have the protonated carbons in aromatic motif. We have non-protonated carbons in aromatic motif, which will show up in the long CP head core spectrum, and we can resolve how polymers are packing with each other. And we can further see that for the CH2 group from the lipid, they don't show correlation with other polymers. So this part is by itself because they self-aggregate to form larger domains. So here are basically the 3 different applications. You can see that we tailor the DMP techniques for different purposes. Basically, whenever you met a problem, you chat with your DMP friend, and then you figure out a way, the best way to solve this specific problem. And it's not a fixed like methodology. So I want to thank my group members who made the research possible. Especially, I want to thank the National Mega Lab who provided me with access to the DMP system and many other high-field MR instruments. So I will stop here and give back to Ruth. Yeah.
Okay, thank you, Tuo. Again, another tour de force and very interesting applications. Very different from chapter 1 from Aaron, but equally good.
We invite you to get to know Q1. Q1 is dedicated to developing NMR systems from 400 to 600, as well as full system upgrades from 300 to 600 as well. You can get a lot of features in a 2-channel NMR spectrometer at a great price. So we invite you to get to know more. Contact us at q1americas.com or info@q1americas.com by email. Thank you.
We're going to wrap up with Galia. Let me see if I can pronounce her last name properly, which I probably can't.
It's Debelucina.
Thank you, Debelucina. I was practicing, but not well enough. She is Associate Professor of Chemistry and Biochemistry at UC San Diego. And she's going to tell us about a different chapter and maybe some things about other techniques and things looking into the future. So Galia, the screen is yours.
Thank you. Thank you very much for the invitation and the opportunity to be here and to share some of our thoughts and recent work on DNP. We are using DNP to study various biological systems, but also in my group, we are very interested in kind of developing different types of DNP, and in particular, with the goal to apply DNP to structural biology in cells. We already had, let me see, can advance. There we go. We already heard from Aaron, really good introduction about what DNP involves. Also, we saw a lot of interesting applications from Tuo's lab. In my group, we are very much interested in proteins. I have been applying DNP to proteins ever since I was a graduate student in Bob Griffin's group, probably starting around 2007. And the way we still do these experiments, we have our favorite protein of interest. We add a biradical, DNP active molecule, usually in large amounts, 10 to 15 millimolar, sometimes even more. We add that to the protein mixture, and then we also have to cryoprotect the protein because protein structure is very sensitive to temperature and also freezing. So in DNP, we freeze relatively slowly, so the protein structure can be damaged in the freezing process, and glycerol helps with that. So we will have our mixture, which is usually very gel-like because of the glycerol. And then we pack it in a rotor. So in my group, we have 1.9 millimeter and a 3.2 millimeter DNP probes. So we can put it in the rotor, and then we can put it in the NMR spectrometer. And so this is our setup. We have a 600 megahertz DNP Bruker system. So the sample goes over here. We can do our regular magic angle spinning NMR experiments under DNP conditions. We have the gyrotron, which produces microwaves that we need to excite the electron spins. And then we carry these microwaves through this waveguide all the way into the probe and then onto the sample. And then we have this very powerful cooling cabinet over here because we do need to do the experiments at 100 Kelvin, which has some advantages and also some disadvantages for biological systems that we can discuss later during the discussion session if somebody's interested. So I like to call this the conventional DNP approach to protein NMR. And so here are just a couple of examples. I got inspired to dig out an old DNP spectrum from my PhD time in Bob Griffin's group, where I was very interested in studying amyloid fibril structure. And I was studying these fibers formed by small peptides. The advantage being that with small peptide, you can introduce isotopic labels where you want them. So I had an N15 label right here, just one, and a C13 label right here, just one. And I was very interested in figuring out if we had this head-to-tail arrangement of the protofibrils within the fibril or this head-to-head arrangement. And so I used DNP enhanced reader experiment to measure the distances between this nitrogen and this carbon. And I did measure distance that was consistent with the presence of this head-to-tail arrangement. The advantage here was that I could do this experiment in an hour, as opposed to conventionally, if I was trying to do this in our regular NMR spectrometers, it would have taken me several days because I didn't have these samples, were very expensive. And also here you need to measure actually several curves, right? DS and DS naught, so that adds quite a bit of time. This is a more recent example from my group. It's actually a collaboration with Francesca Marassi's group at Medical College of Wisconsin. But we were really interested in this membrane protein. It's a bacterial membrane protein called AIL. This protein is actually involved in, it comes from the plague bacterium, and it's involved in the host invasion. And what's really important for its function is actually interactions with the membrane environment. And it's very important to study this in native membranes. So we actually isolated native membranes from E. coli where we were overexpressing this protein. And really the goal here was to understand the interactions of this protein with the membrane. And we had a hypothesis that there are arginine and lysine residues on these loops that are interacting with the LPS layer on the cell envelope. And so we did this NHHC experiment, DNP enhanced, that allowed us to really see very clearly. We have very specific nitrogen chemical shifts for the arginine residue, so they are around 80, and then also specific chemical shifts for the LPS layer. So we have correlations that suggest that these arginine residues are really involved in interactions with the LPS layer, which is very important for the function of the protein. And again, these samples we wouldn't have been able to analyze by conventional NMR because they were bacterial isolated cell envelopes. There's other proteins in there. You can't really pack that much material. And also within the membrane, you also don't have as much protein of interest. So conventional DNP has been very, very helpful in questions like this. And we continue to use it in our group in this context. But what I wanted to tell you today is kind of how can we think about DNP and what role can it play in the next frontier, which is structural biology in cells. Of course, here we are an NMR audience. We all know that NMR can be really an amazing tool for studying proteins in native environments. But if we are to apply NMR to cells, we have to really consider two big problems. The first one is sensitivity. So the topic of today is sensitivity. I tend to think of sensitivity as a physics or physical chemistry problem, where spin physics, such as DNP, which is really what that is, allows us to really boost the sensitivity. And so we can use this tool to help us increase the sensitivity because we only have very few copies of our favorite protein in the cellular environment. And also we can't really pack that many cells within a rotor. So this is the first problem, but the second problem is really the selectivity. So in a cell, we have so many different molecules, so many different proteins. How do we select just the protein of interest within the whole cellular environment and just get NMR spectra of that particular protein? And I tend to think of this as a chemical biology problem because chemical biologists for a long time have thought about techniques that they can use to target specific proteins in cells. Usually their goal is to attach fluorescent probes or control the activity of the protein. But can we adopt some of those tools to develop targeted DNP approaches that allow us to tackle the sensitivity and selectivity problem in one go? So if you were to do DNP in cells, there's kind of two approaches. There is of how to deal with these. So the first approach used quite extensively by Mark Baldas's group, also Kendra Frederick's group, involves actually making the proteins isotopically labeled outside. So you can make them in E. coli, purify them, they're isotopically labeled. But then you can use a technique called electroporation for them to go inside mammalian cells. And then you can have a mammalian cell that is not labeled, but then you've introduced the labeled protein inside. And you can also actually add the polarization agent during the electroporation step, and then that allows you to for the polarization agent to enter the cell as well. So this is one approach, and that gives you sensitivity and selectivity. We wanted to try something different, partly because the electroporation method can be pretty disruptive to the cells. And also you don't really have very good control over how much protein you're adding, but certainly you're adding more than the cell ever uses of this protein. So you're working with really much larger amounts of protein that the cell is used to having. That kind of led us to the idea of targeted DNP in cells, where the idea is that we can find a way to target the polarization agent to our favorite protein of interest. And so we have one polarization agent attached to the protein of interest inside the cell. This idea, of course, of targeted DNP is not new, and I'll show you some examples of how people have approached the targeting aspect. We just brought some. We just brought new tools and new ideas specifically focused for doing this in cells. So if you're to develop targeted DNP, you kind of have to think about several things. The first one is, how do you attach the polarization agent selectively in cells, just to your favorite protein? What enhancements can you expect? Is it going to be useful for what we need to do? How selective is the polarization transfer? Do you polarize a lot of proteins around, or do you know, how big is the range of this enhancement? And therefore, how selective are you? And what are the best polarization agents for cells, which may not be necessarily the same polarizations that we use for conventional DNP. Let's go through these point by point. If you wanted to attach a polarization agent to your favorite protein, there are many ways to do this. The easiest and most popular way is to just take your polarization agent, in this case Amupol, and use cysteine chemistry, whether disulfide or cysteine alkylation, to attach the protein to your favorite, to attach the polarization agent to your favorite protein. Which is great if you're working in vitro. However, this is not applicable for cells because you have a lot of cysteines and you don't have selectivity just based on the cysteine chemistry. The second approach, which can actually be quite selective, and we've seen some examples, for example, in Anne McDermott's group, is to actually take your polarization agent and attach it to a ligand that binds your favorite protein. And so that will ensure that you selectively deliver the polarization agent to your favorite protein. So you get the selectivity that way. The disadvantage is that first you have to have a ligand that you know for your favorite protein, but also every time you want to study a different protein, you have to do new synthesis to a new ligand. So we wanted to be more general than this. And so we developed an approach that's based on genetic code expansion and bioorthogonal chemistry. So genetic code expansion or amber suppression, so the two names are used interchangeably, basically allows us to persuade cells, whether they're bacterial, mammalian, or even yeast cells, to insert a natural amino acids at specific positions in their sequence. And in order to do that, we just need two plasmids and their natural amino acid. We can add that to the cell culture, and the cell machinery does the rest for us. And so this allows us to really put their natural amino acid in whatever protein we want in a very specific way and a specific location. So here is an example of ubiquitin, where we've attached the natural amino acid lysine norbornene. So it's basically a modified version of lysine. So the second thing you need is you need to take your polarization agent and then attach it to a complementary bioorthogonal handle. So we like to use tetrazines because they don't require any additional things. For some reactions, you need copper and things like that. Tetrazines don't need that. And they're really one of the fastest bioorthogonal handles out there. So you can take the tetrazine, and when you put that in a complex mixture, the tetrazine will only react with this norbornene, creating a covalent bond. So you can attach your polarization agent selectively to the protein of interest at a specific location. And this method has quite a few advantages. So it is applicable to any protein of interest. You can do it in bacterial or mammalian cells. The molecular biology is very easy. You can, in principle, work with endogenous amounts of proteins inside the cells. And you can also use it to label things in vitro. And in that case, you can keep the cysteines for something else. So it can be complementary to cysteine labeling as well. One comment, especially when I was submitting this as proposals very early on, was the comment was, "Oh, this will never work for NMR." This in the installation of the unnatural amino acid. However, I think it's one of those examples where people just they've tried maybe once and it didn't work and then they gave up. So we have been able to insert the unnatural amino acid in any protein we've meant to. It's actually the easiest part of all of this. Not every site works. So that's the thing. Sometimes you have to optimize the location of the natural amino acid because some protein sequences, depending on how the protein is processed or how the mRNA is being processed, it doesn't allow the stop codon at certain positions. But generally, if you scan several positions, we've always been able to find a position that works for us. And also we have developed protocols to do this in M9 media, where we're also introducing C13 and N15 labels. So our proteins can be labeled and still have the unnatural amino acid incorporated. So this part, at least in bacterial cells, we feel pretty good about. Okay, so the first thing that we did was we didn't do this in cells, or rather we prepared ubiquitin with this unnatural amino acid, and then we added the polarization agent, purified everything. So we have 100% labeled ubiquitin, 1-to-1 ratio of polarization agent to ubiquitin, and kind of moved on to the second question, which is what enhancement should we expect? And so this is the way we would do conventional DNP. We would take 1 millimolar ubiquitin and let's say add 10 millimolar of polarization agent, and we would get an enhancement of 34. While for the targeted sample, we have 1 millimolar ubiquitin with the polarization agent attached to it, and we get an enhancement of 24. So the enhancement does go lower, but of course we are also using less polarization agent. And what was really kind of important for us to see is that as we increase the MAS, at low temperature doing DNP, the enhancements kind of start to equalize. So targeted DNP may actually outperform conventional DNP at faster MAS frequencies. Okay, so then the second question for me was, what enhancement should we expect? 1 millimolar ubiquitin is great, but that's not the concentration we have in cells. Usually in cells, abundant proteins are often present at 5 to 10 micromolar concentrations. So we want to see how low we can go and still detect reasonable enhancements. So we have here 3 different samples, 3 different proteins at concentrations of about 50 to 60 micromolar, 5 to 10 microgram of protein, and we still get enhancements of between 5 and 10, which still can be very helpful, and as Aaron mentioned, can give us 100 times faster acquisition for experiments that we need to do. So it can make experiments that are not doable, turn them into experiments that we can do within a reasonable amount of time. Okay, and then the next question was, how selective is the polarization transfer? How far does it go? So here is a sample that's conventional sample. We have 1 millimolar ubiquitin with totopol just dispersed in the sample. So here we're looking at the buildup time. So the buildup of the signal enhancement. So you can see that we have a mono-exponential buildup where with a time constant of about 6 seconds. So here it's mono-exponential because the polarization agent can pretty much polarize, a lot of different molecules. When we move on to 1 millimolar ubiquitin with the polarization agent attached to it, we're kind of at the border between mono-exponential and bi-exponential buildup because here the polarization agent can polarize its own protein, but it's also the concentration is high enough that it can also polarize neighboring proteins as well. And then moving on to the lower concentrations, 50 micromolar of ubiquitin. Now we see a very clear bi-exponential buildup. So we have this very fast buildup, which is 0.6 seconds. But then we also have a second buildup, which is about 60 seconds. So we kind of attribute this to two processes. The very short buildup is just the polarization agent polarizing its own protein. And then the long buildup is the polarization agent being able to, through spin diffusion through the solvent, reach a different protein molecule. So this was kind of great to see because as everybody who does DNP knows, what you choose, for polarization buildup time, well, you can control that. So you can have short buildups or long buildups depending on what you need to do. So if we wanted to keep the polarization more local, then we just have to work with shorter buildups. So this was great to see. And then finally, this aspect of how selective is the polarization, what kind of enhancements can we see? So we took ubiquitin with the polarization agent. We actually labeled it in the context of bacterial lysate. So we wanted to have a lot of different other molecules in there. So we have a lysate, about 5% of it is the ubiquitin with the polarization agent. It works out to be about 50 micromolar. And then we looked at what are the enhancements that we get. So the enhancements were not very high. So this is now a complicated system, not a purified labeled ubiquitin. So the enhancements were around 2 to 2.5. But this is actually kind of, at first we were a little disappointed, but then you have to think a little bit more about this. So we have our background enhancement, which is actually the glycerol that we have added for cryoprotection. So that's about 2, while the C alpha enhancement, which corresponds to protein enhancement, is about 2.5. So there is a difference between the enhancement of the background and the enhancement of the protein. And you can do some back of the envelope calculations to kind of figure out your selectivity. So basically, what we, with these calculations, what you can determine is that overall, actually for the ubiquitin, you have an enhancement of about 10. That's your total enhancement, but it's only 5 times higher than the background enhancement. So the selectivity, the enhancement here basically is a factor of 5. So this maybe is not so much, but I do have to point out that here we were using kind of intermediate buildup times. At the time, we didn't go back and do short buildup times. So I think that if we have shorter buildup times, the selectivity enhancement on just the protein that we want can be quite a bit higher. Okay, and then so finally, what is the best polarization agent for cells? So for cells, we have to worry about several things. The polarization agents have to be water soluble, but they also have to be able to penetrate the cells. And then most importantly, they have to be able to survive in cells because cellular environments are actually reducing, which means that our favorite polarization agents, which are nitroxides, they actually tend to get reduced pretty quickly in the cellular environment. So it turns out that if you go from a 6-membered nitroxide to a 5-membered nitroxide, that can help you quite a bit. So in my group, we've worked with these 5-membered nitroxides because honestly, they're just much easier to synthesize. And so it has allowed us to do some chemistry in-house and really kind of do these experiments and proof of principle experiments. We're also collaborating with Snorri Sigurdsson's lab to develop polarization agents that are based on a simple POC. But I do want to point out that obviously this is a problem that a lot of people are thinking about. So from Mark Baldus's group and also Gail Dupep's group, we have these types of polarization agents where we have a tridal and a nitroxide. They're actually really good high field polarization agents, water soluble. But tridal is also has the advantage that it's stable under reducing conditions. And just very recently, there was a paper by Lyndon Elms's group where they looked at gadolinium polarization agents and the stability in cells. Okay, so where are we going with this? So these were all proof of principle, kind of interesting, experiments, but we want to really apply this to some real systems. So we thought that our favorite AIL membrane protein would be a good test system for that because we already have a lot of information about it, but also because it's actually a bacterial protein and it's extra, it's pointing towards the extracellular environment, so we can actually target it from the outside. So we don't have to worry about reduction, and we don't have to worry about how does the polarization agent go into the cells. And so just last week, actually, my student Nesrine Eletram, she gave a presentation at the Global NMR discussion group. So if you're interested more in this project, we're still working, finalizing some things. But the idea here is to we've switched the amino acid. Now we're working with more efficient natural amino acids. We have slightly different versions of the tetrazine. And then also we've tried several different polarization agents. So this particular one was synthesized by Melinda Serrato, but we also have a polarization agent now from Snorri Sigurdsson's lab, which has higher enhancements, which we're developing for this system. So, so we hope to use this targeted DNP to understand the interactions of this protein, not just with the membrane environment, but also with some human proteins that are involved in the infection process. So here's my summary. So I think that targeted DNP is a promising technique to study proteins in complex mixtures. And we have really worked out a few of the technological aspects of this. I think the unnatural amino acid incorporation, I feel really good about it. In bacteria, we have some promising DNP enhancements. There are obviously a few things to optimize, and we are hopefully going to start working in mammalian cells soon with this. But also, I think there's already, a lot of room for interesting applications, even with the information that we already have and the technology that we already have. Okay, and then finally, thank you to my group. So a lot of the targeted DNP work, early targeted DNP work was done by Bryce Ackerman and Byung-Jun Lim. And then also the recent DNP work has been done by Nesrine, and our undergraduate, Drake Jimenez, has actually spent a lot of time figuring out how to install unnatural amino acids in his favorite protein. So I'm ready to take questions, I guess, or everybody can take questions now.
Okay. Thank you, Galia. Oh my goodness. Okay. So we are ready for questions. So the screen is open, I guess. So please you can put something in the chat and we'll call on you, or you can raise your hand and we'll figure that out. And of course the panelists should feel free to ask each other questions. And if I don't hear anything soon, I will start it off. One of the things that I heard from several of you was the the issue of selectivity in different contexts. And I have a little trouble thinking about that because often you want selectivity, and I don't always know whether to think of a layer or a surface layer as a barrier or as an opportunity to select what you're looking at. And I'd like to have some help from the panelists in thinking about that. So I'm thinking about Tuo's example with the lignin, or I'm thinking of Aaron's example of all of these surfaces where the surface is what you want. And there may be cases where what you want is the thing inside, further in, and then you have to design things differently. So maybe people could talk a little about that issue and help me out.
Sure, I could add a quick comment. So, yeah, I think it's really dependent upon the context of your applications and the types of problems that you're trying to solve. So in material science, in catalysis, we're often interested in understanding the surface structure of the materials. Now, there are definitely examples in material science where we also want to study the bulk structure. So if we think about solid state NMR, lots of people are studying glasses and polymers and trying to uncover the molecular structure of those materials. And there you'd like to have your polarizing agent kind of homogeneously dispersed. So those would be examples where you don't want kind of surface selectivity. Another case where that comes up is in pharmaceuticals. So if you think about, solid pharmaceuticals, those are usually microcrystalline solids or again, drugs blended into a polymer. And there we'd like to be able to do DNP on the bulk. I could show a slide really quick to talk about maybe your question, Ruth. So,
Sure. Yeah,
and maybe a detail that I skipped over when I brought up the example of histidine.
Okay.
So, when we do experiments on microcrystalline solids like histidine, what we will do is just take these kind of microcrystals that are, we just get them in a mortar and pestle, we grind up the material, but those crystals are still pretty large, they're a couple microns. But it turns out that if you have organic solids or organic material and you have the radical restricted to the surface, you can study the equations for kind of bulk spin diffusion based upon a kind of Fickian analysis of the diffusion process. And so this is a kind of mathematical model that shows that if you have a radical restricted to the surface of an organic microcrystal, even if your microcrystal is 4 microns in diameter here, you're still able to get an appreciable amount of proton spin polarization pushed into the core of this material. So even when we have kind of larger objects that are organic, as long as we have a network of abundant spins, which are strongly dipole coupled enough, we can push the polarization into the interior.
Right. So, but you need, as you just said, you need to have a good spin diffusion for this game to work. And rigidity is helpful as well, right?
Well, rigidity is it's long T1. That's the key.
So, okay, fine.
Your
however
diffusion,
you get it, okay.
Well, your diffusion length is square root of 6 times your diffusion constant times time, and so time is ultimately limited by T1. So the longer your T1, the longer you can kind of push polarization over length scales.
Right.
So that's something to think about there.
Yeah. So I also have a quick addition here, so the polarization heterogeneity, so is something I think we cannot avoid completely at this moment, but it's basically how we can use this to solve the problem of interest. Yeah. Like the surface and also interaction interface.
Yeah. I know I've asked this question to you, Tuo, before, because well, I mean, if you can't avoid it, at least if you know about it, there are going to be cases where I mean, I don't know. I think another way to try to attack it would be to design a radical or do people use combinations of radicals? I don't know. Where one would suit one situation, another with another. I don't know. That may be a little nuts, but
Yeah.
Just asking.
Yeah. So another thing I want to mention, especially to the plant samples, is if we think about the lignin, so when we have small lignin particles like nanometer scale, very small, then they typically can be polarized quite well under DNP. So they show similar enhancement as the carbohydrates. But in some really old trees, where the lignin become large and layered, so then we have issue polarizing them very well. And if you think about lignin is hydrophobic, even water cannot diffuse very well
Yeah,
into
so it's
those
hydrophobic
clusters.
and it's proton poor.
Proton poor, yeah, so that's really
Add
challenging.
insult to injury, yes, I
Yeah,
see.
yeah. And the radical dissolve in water, they cannot even get close. So, so for those things, it's really challenging. Yeah.
But why don't you swell it with organic solvent and take something like Tecpol, which is going to be very, probably partitioned into more aromatic region.
Yeah, we can do this. The issue is the reverse will give question is not like a native condition, so no longer like a water base, so yeah, but that's something to try, I think. Yeah.
I was just
ahead.
gonna add one other comment. I think one other thing that people are using to maybe in more materials examples to polarize the bulk, like in glasses, you can introduce paramagnetic metal ions, so gadolinium. In some cases, manganese works okay. Copper, so like Michaleskis' group is looking at materials that have these intrinsic metals.
Yeah.
And then we've had a little bit of success in both pharmaceuticals and some inorganic systems in using gamma irradiation to create radical defects that can be used for DNP. And that would also let you polarize the bulk because ionizing radiation is very penetrating. So when it does create radicals, those tend to be kind of homogeneously distributed.
We have a question in the chat. Andy Lipton, do you want to speak up and pose your question?
Had to find my mute button.
Please.
Hey guys, all of you, nice talks as always. So we've got the high field 800 megahertz DNP here. One of the challenges I had when I was running some test samples on it, I was never really getting a good optimized DNP enhancement. And I suspect that we were optimized for a standard radical, and everything I was trying to use was probably out of the range. I mean, Aaron, I think you mentioned, put some endogenous radicals and do you move the field a lot to find those, the optimal enhancement? I mean,
yeah, so I think for most nitroxide biradicals, your main magnetic field is set such that your microwaves are going to irradiate around 2.0045 G value. Okay. So if your radical has a significantly different G factor, which when you look at things like gadolinium, those are below 2.00. So you end up having to sweep your field by several thousand ppm. That's pretty common if you want to work with something like gadolinium. Some of the irradiated materials we've studied, those have had radicals that have G factors closer to like 2.002. So we end up having to sweep the field by about 1,000 to 2,000 ppm. Also, even just those mixed tridal or BDPA nitroxide radicals, again, I think you have to sweep the field by roughly 1,000, 2,000 ppm. So there's definitely significant field sweeps. And then, I think a good general thing to do is, as NMR people, when we do DNP, we have to learn a bit of EPR. So we have to start doing X-band EPR. And even better, go to Tallahassee and use the facility there and get them to run your sample at W or D band, because the higher you get in field, the closer you're going to get to the field that you're using for the DNP experiment, and the more representative your EPR spectrum is going to be for what you're going to see at high field.
So those that was my thought. We just have to get in the habit of running the EPR first and find out where we're at.
Yeah, I mean, X band can be helpful, but if you're dominated by hyperfine couplings, those are just going to dominate the spectrum at X band, whereas G anisotropy tends to dominate at the higher field. So it's also really valuable to get on a very high field EPR somewhere, especially if it's a weird radical that has not been studied before, right?
Right. Yeah. Thanks.
Great question, Andy. Thank you.
I mean, I think in a way, particularly this last answer underscores the need to be empirically bold in there's not a formula really. I mean, there are common things, but there's a lot of optimization, and you just have to tolerate it. I mean, there is some element of black art, as some have told me. I think Bob Griffin made that comment to me pretty recently, that is inevitable because each case has its own wrinkles, if they're interesting cases anyway. So I think that's an important general kind of guideline, because it's not just that you have to optimize and be patient if you don't know what you're doing. I think even if you do know what you're doing, you must do that. So I don't know if that's disappointing, but I think it's true. So yeah, good.
I think I would just comment, Ruth, I think that's like an accurate statement, but also, as you get experience with this technique, it's like anything else, you can make educated guesses,
Yeah, sure.
How big are the particles of the material I'm studying, or is it hydrophobic or hydrophilic? What types of radicals should I use? I mean, for me, one of the biggest things that we find is a problem, and it's a general problem when we do solid state NMR. Solid state NMR, T1 can be anywhere from 10 milliseconds to 10 hours. And you almost never know. I shouldn't say you never know, but sometimes it's very surprising, the first time you put a sample in the magnet, that it turns out to be much longer or much shorter than you think it's going to be. And a lot of DNP is T1. It's kind of reverse from normal NMR. In some ways, we like to have a long T1 when we do DNP, because that ultimately allows us to bring the spins out of equilibrium in a positive sense, hyperpolarize them, and get better DNP enhancements.
Yeah. Well, if you're not thinking about T1, you'll lose more often than you'll win, I think. And particularly in solids, you have to think about it all the time, I think. Maybe in, it's a little different in liquids, but yeah. Okay, cool.
Can I ask a quick question? This
please.
Is Krish. I was intrigued by Galia's introduction on polarization in cellular level. I was quite intrigued. I'm going to ask a question, but before I ask that question, I want to remind everyone I'm coming from a world where if you can't pipette your sample in and out of an NMR tube, it's hopeless. So that's my preface before I ask this question. How far are we, with some of my experience in the pharma world, how far are we in being able to do these kind of extend this cellular polarization on cellular level to actually in vivo, in image-guided spectroscopy, at least in animals? I guess this is a very common thing come to. I told you that it's a crazy question. To put it in context, these are the kind of questions that came to us when I was in the pharma world, from the biology group. What
Mhm.
can you do for me? Because I have this animal model and I want to understand this. So, how far are we? Maybe 10 years, 15 years, 20 years, technology, impossible?
I mean, I don't know about animals. I think we are still quite far off from working in live animals because that means all of us also have to really learn MRI or how do you work with something that's not 1.9 millimeters but big. So there's a lot of things to think about there. But I think for cells, working in living cells, right? Already solution NMR, there's amazing advances that people have made in like studying proteins by solution NMR in cells. For solids, I think the biggest issue is how much the magic angle spinning can affect the cells. And obviously, if we want to use DNP, we really have to kind of adapt these more, think about Overhauser DNP or think about some hyperpolarization methods, borrow them from solution NMR and from MRI to think about living cells. So I mean, but I think that's probably not that far off. I think really, we already have a lot of advances. The challenge is, to pull something off like that, you have to really pull from a lot of different techniques. You really need to be also a cell biologist, you need to be a chemist, you need to be MRI, know everything from, MRI, EPR, solid state NMR, solution NMR. So there's just that, I think that's and be able to actually build the instrumentation and adapt the instrumentation. But I think it's also a fun challenge to think about.
Yeah, I mean, I am impressed with the way this technology has moved forward so fast, relatively fast in my time. In my time, so I wouldn't be surprised if before I leave the world, I will see something in that level. I'm very impressed with what is going on. Very impressed. Thank you.
It's certainly a very exciting part of our field, and it is challenging. I think that's fair, but certainly now that there is commercial equipment and or national facilities that have these things, that really opens it up, and that's that will help for sure. Okay, I think we are kind of at the end because I don't see other questions. So I want to thank our panelists for being willing to do this, and our listeners and other participants for being here, at least virtually, and close it up and say we're done. Unless Krish has something else to say.
Yes. Before I go, I want to thank everyone, thank the panel. Wish you all a very happy holidays and Happy New Year, and see you all in January again for the next workshop in hyperpolarization, a subject that we all would be interested in to hear from Pete. Patrick, thank you. I see you all.
Stay safe.
Thank you very much. Yes.
Okay. Thank you, everyone.
Thank you. Yeah.
Bye.
Bye.
Thank you.